Hey, everybody. So my name is Josué Ortega. I'm a postdoc in the lab of Jess Cardin and Dave Van Dyke. Today, I'll be talking more about spatial temporal data and what we can do with it, and also multimodality in the context of spatial temporal data. I'm surprised that this slide has not come out yet, but I missed the morning session, so I wonder if that's, I missed those. And one thing I think we're all interested in, everybody has talked about, is that there's processes in the brain that happens on multiple scales, both in space and time, right? If we think about things like memory, we have things that happen on the molecular scale, like on the boutons level, then all the way to the circuit structures that are changing. So I think we're all interested in finding models and data that are able to integrate all these modalities, able to understand how these processes happen across scales. And at the same time, as people have seen in this workshop and along other places, I think a lot of models kind of are in two different axes, and one of them is kind of these high predictive power models that people have been talking about, where you can include a lot of data, kind of compress the data and be able to predict a lot of things out of them. And then the other subset is this hypothesis-generated model, which are models that are more tailored to specific applications or specific scenarios and conditions that then leads to the hypothesis can be tested experimentally. And I think a lot of people in this work are trying to get to this kind of third stage, the orange, where we have high power models, but at the same time allow us to generate hypotheses. So what I'm going to present today is kind of two case studies of two projects that are one in the first scenario, which is a very classical foundational model for brain data, as we have been seeing, and a second one, which is my work that tries to find a way of kind of have hypothesis-generated models at the same time of having high predictive power. So the first one is called BrainLM. We were trying to find a data set that was very large, equals to the EEG data. And what we found was fMRI data. We were trying to study spatial temporal data. And this is the UK Biobank data set, which some of you might know. It's a 40,000 individuals recordings where you get an fMRI of each of the patients. Sometimes it's resting state, so the person's not doing anything. Sometimes they're performing a task with like scene and image. And at the same time, we get medical questionnaires. So we get things like age, if they have any kind of depression, scores based on some very standardized kind of questionnaires and things of that sort. And what we really wanted to do here is kind of test this idea of the foundational model. Grab all this data, pass it through a model, compress it, and then try to use it to predict then later clinical variables. So the setup that we use is one that is, I think, very familiar for our second day kind of workshop, is the mass autoencoder training. So one key component of spatial temporal data is that there's correlations in space and time. And one kind of more in the industry setting where one might want to think about that is video. So in video, you have pixels nearby, they're correlated to one another, but then the pixels are also correlated in time. So what we find actually for trying to model these type of spatial temporal data is that the level of masking required to train such a model and be successful is really high. It's above 75% to 90% of the data needs to be removed in order for you to get a successful model in spatial temporal modeling, kind of mass autoencoder setting. And the way we did it was similar to a lot of other work, random masking and also future masking prediction. And what the model, this is an encoder-decoder architecture where only the visible tokens get passed through the encoder, and then the masked tokens get passed through the decoder as well. And what the model is required to do is just reconstruct the data that is missing, given the remaining information. And then finally, what I'm going to show is an application specifically about predicting clinical virus, given fine-tuning's, let's say, second stage, using this foundational model. So first, scaling law plot, I think that's required at this point. We have a number of parameters here in different colors, and then we have the reconstruction R squared. This for a model that has 80% masking, so that means 80% of the data has been removed. And what we find is that with increased number of parameters in the model, plus increased amount of data, so this is 100% is 40,000 subjects around roughly, without this removal of held out. So this performance is held out performance. How well can you generalize to new subjects given training on some percentage of the data? So what we find is that the generalization capabilities improves of the model with more data in training. And this is kind of an example visualization of that. These are reconstruction of held out UK Biobank data. And the model you can see in red is actually pretty good performance. And then this is an even external validation is the Human Connectome project, which is another fMRI data set, which is completely separate from the UK Biobank. And we still can do the reconstruction of all subjects in the UK Biobank, which is around 1,000, with pretty good performance. Sorry, the Human Connectome project with very good performance. And then kind of the next step is now we pre-train the model, it performs really well. And now we want to see like how well can we predict these kind of clinical variables. This is a regression problem. They get a questionnaire and they get a score based on their depression level or neuroticism or anxiety or age. And this is kind of our models, BrainLM, two ones which are in pink and brown. And we find that outperforms other models in terms of performance, but the metric is still can be improved. There's still, I think we need to do a qualitative jump, as people were talking in the panel before, where we're kind of improving little by little the performance. And the question is there an amount of data where which we can do a qualitative jump where we really improve the performance on all these models? And I think we still have a lot ahead performance that we can improve, at least in the fMRI context. So kind of a summary of BrainLM is that we have not yet found a plateau. This is not a task that has been solved. There's definitely plenty of space to be built, better and better models for fMRI. We find that massive encoder training seems to be a successful pre-training strategy for spatial temporal data. But as I said, I think the level of masking is a parameter you really have to play about, given the large amount of correlations in the data as they presented. And another thing that is important for fMRI is that the qualitative data, even though it's standardized in the tests like anxiety score or some sort, these are question surveys, so people lie, or they think their perception is different, so therefore the questionnaire is very noisy. So I think we're trying to also find high quality metadata that we can use for these sort of tasks, because the UK Biobank can be very iffy about the data itself in this metadata component. I think they're better standardized in the fMRI side than on the metadata side. So now, case study 2, this kind of intersection between two things. And now we're going to mice. And you will say, human mice? Well, the reason is that we have so many techniques to kind of study multiple things in mice, right? We have like, sort of like electrodes we can put them, techniques like optogenetics, we have calcium imaging, we can do genetic manipulations, all sorts of things. So here, compared to, I think, everybody else who's kind of focused on very fast tasks, I'm focusing now on learning, a very slow process. So what we're doing with the mice is that we're training it to do a delayed visual detection task. So the mice sees grading with a specific contrast, which is 100% at the beginning, and then it has to lick when presented with stimulus, and if it licks during the presentation, it gets a reward. So a pretty simple task. And kind of this task has 4 different separate periods. One of them is just learning to lick. The other one is presented with the visual cue, licking. The ITIT punishment is when the mouse licks, when there's no stimulus, it gets penalized with a longer wait between trials. And the mice really wants to get those trials small because it's water deprived, so it really wants to get as much water as possible. So it's incentivized to decrease the ITI. So really, in the ITI period, you want to decrease false alarm and keep your correct rate high. And then finally, we have a psychometric period where the mice, we change now the contrast level of the stimuli, and the task becomes much more difficult in terms of detection of the stimuli. So here you want to keep your kind of correct performance goes down because you're no longer detecting the stimuli, and your vision has to become more attuned with lower contrast stimuli. So this is kind of an example visualization of the mice. It's on the wheel. You'll see the grading soon here. It should, okay, the grading. And then the mice starts moving, and we kind of licks as this is stimulus. This is the correct trial. And at the same time, through all the days that the mice is learning to do the task, we're recording multiple things. So this is a technique developed by Sweta Lohani and Animo Bernie in Jess Carding's lab, where we have 2 different indicators for different kind of signals. So if you can see this is a dorsal cut of the brain. This is 1P calcium imaging, so white field. This is the front of the brain, which we like the whiskers. This is the back of the brain, which we like the visual cortex. And we have calcium indicators, which is kind of a proxy of neural activity. And we have also neuromodulatory signals, acetylcholine, which has been shown in many research to be involved, I guess, in the teaching signal in terms of learning. And at the same time, we're recording things like face video. We have the face motion, we have locomotion, pupil area. So what we really want is to be able to integrate all these signals into a model and be able to understand how are these dynamics changing through learning, and what can we learn about the relationship between these signals as learning is happening. So no shocker, transformers. Next transformer talk. So what we did is create this kind of multimodal transformer for integration of different modalities, where we have the calcium modality, acetylcholine modality, and then kind of these behavioral modalities. And what we did is kind of break the signal similar to what they do with video kind of transformers through small grids of tokens and kind of represent different areas as different tokens into the system and create this space. Well, technically this is no space, but modality like red, blue, but there's different spaces in each modality. And we have 3 types of kind of masking strategies that we're using for pre-training this multimodal model. So one of them is random masking. Remember, for this, we have to use very high in the previous scenario. Here, I use up to 90% masking in order to train the model. If you use any less than that, you get a model that literally tells you absolutely nothing about the system. Then we have forward masking, where we use, we're sampling the data 10 hertz. So we use 400 milliseconds worth of data to predict 1 second worth of data in the future. So we're doing a lot of forward prediction. And then finally, modality masking, which is we remove one of the modalities and try to predict it from the other. So similar to the last 2 talks about conditional generation given other modalities. And what we're using here is the data from the visual stimulation time point. So when the stimulus gets presented and what the mice does given that context. So first, here is some kind of very clear, some benchmarks of our R-squared predictability in terms of future predictions. So remember, we're predicting 1 second worth of data in the future given 400 milliseconds. And we find that this MMT model outperforms other models, even with the same input into the system. I think there's something interesting about transformer and modularity in architecture that allows to add different things into the model and keeps kind of modality independent information and share information in kind of separate space. We're still trying to explore that idea. But when we do that with, like, I don't know, SSM or GRU, where things get smashed into the same space, sometimes we find that the model tends to have a harder time when more modalities are added into the system, where compared to modality-specific information. And that's, I think, why some people have generated these kind of more modular SSM models or like other architectures that are more modular. But I think transformer kind of, you get that modularity sometimes for free. So we're still trying to explore why that's the case. But what is interesting is that when we do that predictability in the future and we just kind of assemble it, we see that the main point is that the MMT model basically can predict into the future with consistent performance compared to the other 2 models, which basically decay performance as time goes more forward into the future. I see we're also trying to explore how far in the future can we push this? Like, can the model really get to like 1 extra second in the future? Like when it starts decaying and under what conditions? We also find that predictability is dependent on the certainty of the mice. So stimulus, they're lower contrast and they're harder to predict. They're also harder to decode the neural dynamics. So I think uncertainty in the dynamic itself actually tells you a little bit about what makes it easier and harder to predict. Very certain trials, much easier to predict compared to low confidence trial. And then similar to 2 talks ago, we're trying to use attention to try to understand the model. So this is kind of the second component of interpretability. We're trying to see, like, what can we extract from the model and understand learning given the context of a very high performing model. And this is kind of an interesting finding. So here, what we're trying to do is we add an attention head to the model, a readout head, and try to predict correct versus false alarm files. Remember, during the operant training period, the mice is learning to withhold its licking in order to perform correctly and pass to the next stage. Otherwise, it gets penalized with a longer ITI. So this is kind of the training objective of the mice during this learning period. And what we find, which is interesting, is that if you look at the attention of the calcium versus acetylcholine, you see things that are kind of expected. You see attention in the visual cortex, retrosplenial cortex, with kind of a little bit higher visual areas, and also kind of some preparation, and then frontal cortex and motor cortex for decisions. It's a very classical, it's a bunch of different areas and kind of all the brain light up. Similar in the psychometric period compared to correct versus incorrect trials, though there's more on the retrosplenial and visual cortex. However, the biggest difference is in the acetylcholine signal comparing operant training compared to psychometric training. And what we find is that there's no attention to the acetylcholine signal in the psychometric period compared to the operant training. That means that even though we have two separate modalities, the model is able to pick up modality-specific information relevant to learning, given the context of classification between correct and false alarm trials, which is kind of what the model is waiting for. Then we look at the more detail on another specific question, right? Once we go to the psychometric period, what the mice is learning is no longer to withhold living. They already learned that in the operant training period. They already got good performance. They passed to the psychometric. However, what it becomes difficult now is the 2 to 5% contrast level, which is really near the perceptual threshold of the mice. If you compare and you break down the attention that the model is paying to near threshold compared to high contrast stimuli, what we find is that, again, the acetylcholine signal shows, but actually in frontal cortex, there's no longer no attention in visual areas. That means that the main thing to differentiate correct versus incorrect trial, which is the thing that the mice is learning in that period, it's actually probably evidence accumulation. You have to wait a little bit longer in order to make a decision because evidence is so sparse, right? Contrast is a little bit lower, so you have to see, it's like, is there actual stimulus there or not? And similarly, in calcium, we find that there's frontal cortex, retrospinal, actually less visual cortex in this case compared to the operant training period. So kind of what we have found here is that first, master encoder training seems to be, as other people have shown now, is a successful method for multimodal data sets. Even modalities tend to be very different. Attention rollout seems to be interesting enough to find differentiable effect in the acetylcholine-specific channel during operant versus psychometric training period, kind of indicating what things that we can explore later in specific kind of scenarios in more experimental setting. Like, can we look at, for example, frontal cortex during learning of the stats, specifically in the psychometric training period? What can we learn about what the system is doing with that? And then one key component that I think is a kind of a caveat here is that all-to-all prediction decreases interpretability. So we try to do a version of the model that had try to classify correct versus incorrect versus false alarm versus the mice didn't do anything when no stimulus got presented. And when we did that, the attention was way less interpretable. We're trying to get kind of specific motifs for classification. We find that this pairwise comparison was actually better for kind of generating interpretable attention in condition-specific setting. So to end, we kind of have this kind of lessons and questions. We haven't found a trade-off yet between performance and interpretability. I think there's ways that we can push both at the same time, which I think is very interesting. I think if you follow Memming's idea that I'm interested in understanding the brain. I don't really care that much about performance. I want a mechanistic understanding of the brain. Then I think it's interesting to think about how we're setting up these objective functions. Like now we're using mass encoder, but there's a very interesting component about which is what they call, I think, temporal contrastive learning, where they're trying to compare the distance between pairs of recordings and how far away they are. And I think definitely a lot of this work has shown that we need more additional methods for multimodal and multi-scale training. I'm pretty sure that master encoder training is not the way to solve this problem, but it's a good step to start going that way. And I think we need to come up with new objective functions. So finally, I want to thank, these are kind of two projects from my two labs from the Van Dyke lab, Antonio Fonseca, who's my co-first author, Syed and David, who have been there to support on the project. And then on the Cardian lab, Sweet Lohani, who collected this beautiful data set that I've been using for the last 2 years. And then Hannah, who is my experimental collaborator, who's now validating the stuff that we find on this paper. And then Jess for kind of all the support in her lab. Thank you.